Hello everyone, our team for Senior Design 2 is BioNerve. The group members are Mariam Harani, Anna Hodgson, Tom Yang, and Ryan Gubert. Our mentor is Dr. Yang from the University of North Carolina at Charlotte. The objective of our project is to design an in vitro model for how low oxygen concentration, or hypoxia, affects an isolated part of a neuron. The scope of the project includes creating the hypoxic media, developing a chamber system that can house and isolate specific parts of the cell, measuring the oxygen concentration of the media, creating PDMS microchannels, plating the cells, and the overall experimental design. The most intricate part of our design is how the cells grow through the microchannels underneath the 3D printed housing. As seen in the diagram, the cell culture is plated inside the open chamber before the lid is placed on. Before that, the bottom part of the chamber is bonded to a thin layer of PDMS, which was poured over a mold to create the microchannels. For viewing purposes, the bottom of the thin layer of PDMS is then plasma bonded to glass. To create the hypoxic media, an incubation chamber is used in conjunction with the vacuum and nitrogen gas. Inside the chamber, when the vacuum is applied, oxygen leaves the media because of a pressure difference. Nitrogen is then pumped into the chamber to replace more dissolved oxygen. A syringe pump is used to slowly push the hypoxic media through the axonal side of the chamber. The diagram shows how the axonal side and cell body side are connected by the microchannels. Because of the size of the channels and that the cell body side is open to air, the barrier between the two conditions is maintained throughout the experiment. The PDMS is created by mixing two chemicals together and pouring the result over a pre-made mold. The PDMS then takes on characteristics of the microchannels. The PDMS must be thin, however, so it is spun in a digital spin coater for 20 seconds until evenly spread over the mold. The PDMS and the 3D printed housing are bonded together to create a leak-free seal. 10 of the completed assemblies will then have small tubes attached to the input and output of the axonal side of the assembly. Hypoxic media will flow through the system from a syringe pump operating 10 syringes simultaneously. An oxygen sensor will be placed at the output and will measure the oxygen concentration inside the chamber. The cell cultures will be placed in a large incubation chamber that is temperature controlled. The experiment will run for 7 days and one cell culture per day will be removed for viewing in the microscope to observe the effects of axonal hypoxia. Our team expects this method, as well as a chamber design, could be utilized for testing the effects of medicine or other cellular conditions on isolated parts of a neuron, furthering research on diseases causing neuron degeneration or death.